Good afternoon, everyone, and thank you for inviting me. Well, this project was carried out in collaboration with the Universidad Nacional de Colombia, the Pontificia Universidad Javeriana, the Institute for Legal Medicine of Innsbruck, and it was grant sponsored by Colciencias. Here, I will be talking about topics related to the peopling of the Americas by anthropological perspective. In this context, two specific variables, the shape of the skull and the dental size, are considered. As for the shape of the skull, we have the dolichocephalic type that are elongated skulls, characteristic of Paleoamericans, and the brachycephalic type that has a more rounded shape and is characteristic for recent settlers. In terms of dental size, the classification is divided into macrodonts, which are large teeth, and microdonts, which are small. Different peopling hypotheses, such as migrationism and microevolutionism, have been formulated based on these two variables. These two differences are portrayed by the oldest Paleoamerican throughout the American continent. This map here shows the geographical location of some bones remains with Paleoamerican morphology. One of Colombia's most studied archaeological sites is found in the Eastern Andes. Some of the most found remains in our country have both types of morphology. The oldest remains show a dolichocephalic morphology, while the ones from posterior periods have a brachycephalic morphology. Based on these findings, it has been to classify these remains in different archaeological periods. The early Preceramic period is characterized by dolichocephalic morphology and large teeth. Individuals from this time were hunter-gatherers, and the archaeological sites are Chequa, Tequendama, Nemocón, and Floresta. The late Preceramic period is also characterized by a dolichocephalic morphology, but with small teeth. Here, hunting, gathering, fishing, and horticulture were the predominating activities. Remains from this time have been found in Aguasuque and Vistahermosa. The next period is the formative or Herrera, in which the brachycephalic type morphology and cranial deformation appear for the first time. During this period, people started farming and congregating around a small village, and inside pottery can be found. The archaeological sites are Madrid, Laguna de la Herrera, Sogamoso, and Duitama. The early Mooskaa period is characterized by brachycephalic morphology. This is considered a transition period in which the painting ceramics also appear. The archaeological sites are Candelaria, Porto Alegre, and Sogamoso. Finally, in terms of the late Mooskaa period, there are brachycephalic morphology, intensive agricultural activities, and the societies became hierarchical. Remains of this time have been found in Bogotá, Tunja, Duitama, and Sogamoso. Considering these findings, it is important to begin to enrich hypotheses about the peopling of Colombia with the result obtained using genetic tools. One of the most useful genetic tools for reconstructing ancestry is the mitochondrial DNA, and there are four main reasons. High copy number, which means that large amount of mitochondrial DNA can be obtained. Maternal inheritance, which provides a more direct genetic lineage. It is not recombining, and it has a high mutation rate. Based on this result, from mitochondrial DNA studies, it has been possible to map the world's human people in pathways. These pathways are represented by continent-specific haplogroups. In the case of the American continent, 5 main haplogroups have been found: A, B, C, D, and X. A region that allows us to perform both temporal and spatial population genetic analysis is that of the Sun Temple in Sogamoso, as it is located 210 kilometers from Bogota. This site was a religious center of major importance for the Mooskas. In this place, bones from all 4 archaeological periods have been found. Bone remains belonging to the pre-ceramic period dating from around 8,000 years before present. Remains from the formative period dating from around 2,000 years before present, and remains from the early and late Mooskaa period. These findings have made the region ideal for temporal spatial analysis. For this reason, our main purpose was to analyze ancient skeletal remains from 3 consecutive cultural periods found in the surrounding of the Sun Temple, to shed light on their population dynamics and origins. To complete this purpose, we worked in collaboration with Dr. Walter Parson and the Institute for Legal Medicine of Innsbruck, and concluded the sequencing of the complete mitochondrial genome. We analyzed a total of 45 samples belonging to the late Mooskaa, early Mooskaa, formative, and pre-ceramic periods. Mitochondrial haplogroups were defined, and we obtained these frequency distributions. In the Mooskaa period, we found haplogroups A2, B2D, C1b, and D4H3A. Within the most prevalent haplogroups A2, subhaplogroups A2AC, A2AD, and A2Y were found. In the formative period, we found haplogroups A2AC and B2D. In the pre-ceramic period, was obtained results for 2 individuals, one with C1b and the other one with C1b8. A network analysis was performed based on information from the mitochondrial DNA control region, which showed that there are some shared haplotypes. In haplogroup B2D, there are shared haplotypes between the formative and Mooskaa periods. And in haplogroup C1b, there are shared haplotypes between the pre-ceramic and the Mooskaa period. A comparison with the other population was performed. This comparison showed haplogroup A2AC as the most frequent in our sample, and it is highly present in modern Colombian populations, including the Embera Chamí, Vaupés, and Bogotá. This haplogroup is also shared with population from Panama, Venezuela, and to a lesser extent from Ecuador and Peru. Haplogroup A2Y had previously been reported in an isolated Waurani population from Ecuador and was thought to be unique to them. Another important haplogroup is A2AD, which has 3 clearly differentiated clusters. The last cluster is characterized by a polymorphism in the 60,259 region that led us to consider it a new lineage. This haplogroup has also been found in contemporary population from Panama and the Dominican Republic. In this phylogenetic analysis, our samples from the Mooskaa period are shown in red boxes, and samples from the formative period are shown in orange boxes. We found that 8 of these samples belong to the A2AC1 lineage and form a structured subclade characterized by a transition in the 9591A region shared with 2 contemporary mitogenomes from Colombia and Bogotá, and Cuba. These findings leads us to consider this subclade as a new lineage. The coalescence age for the new A2AC1 calculated as 4,000 years. Additionally, in haplogroup A2AD, there is evidence of the new A2AD lineage, and the coalescence age for this new subclade was almost 10,000 years. One complete mitogenome from this study, belonging to the Mooskaa period, shared with 2 extant Ecuadorian Waurani haplotypes that had defined haplogroup A2Y. The coalescence age for this lineage was calculated as 10,000 years. In terms of haplogroup B2, when analyzing sublineage B2D, we found that 2 mitogenomes from this study, one from the formative and one from the Mooskaa period, were identical with 5 modern Colombian samples. The coalescence age of B2D was calculated to be 7,000 years. Haplogroup C1b was the only complete mitogenome from this study. This haplogroup belonged to the Mooskaa period and could not be assigned to any described subclade. Haplogroup D4H3A from this study, also from the Mooskaa period, fell into an unnamed clade defined by 2 transitions shared with 2 extant samples from Mexico and another sample whose origin is unknown. Dispersion analysis of mitochondrial haplogroups from the MPOP database confirms the high frequency of haplogroup A2AC in northeastern South America. Shows that haplogroup A2AD is scattered in Central America and present lineage A2Y as restricted to an isolated population in Ecuador. On the other hand, haplogroup B2D is shown in 2 focal points, one in Central America and the other one in Colombia. Lineage C1b is currently spread throughout the continent, and C1b8 has been reported in California, Mexico, Venezuela, Ecuador, Chile, Argentina, and Brazil. Finally, D4H3A is a rare founder haplogroup, first described in Ecuador, with currently mainly spread along the Pacific coast in South America. In conclusion, this first complete mitogenomic report of pre-Columbian human samples from Colombia demonstrates how ancient DNA samples can contribute to the reconstruction of a more complete picture of the past. The novel ancient mitogenome from this study, belonging to all 4 major clades of Native American founders' mitochondrial DNA lineages, had highly desired data for reconstruction details of human migrations. Unlike other ancient mitogenome studies, we reported polymorphisms beyond the haplogroup nodes, thus contributing to more precise phylogenetics and convergence estimates. These samples set clearly shows, at least for the maternal side, that despite European conquest, the genetic heritage from the original human settlers of the Mooskaa territory is preserved in today's population at a high degree. Finally, I thank you. Thank you for your attention, and thanks to all research partners and institutions. Thank you.